All right, I'm Chris. I'm a trauma surgeon in Nebraska, and I work closely, I think, with several of you. A lot of what I'm going to show is results of a number of collaborations with the Patch Group and Gene Moore from Denver, as well as Marty Schreiber, who's not here. So the topic is the role of fibrinolysis and antifibrinolytics, which I think you've heard a lot about, including a lot of the trials, in complement-mediated inflammatory responses after injury. And it's kind of a longer, broad program that I've been working on since I was a postdoc at MIT. So disclosures, we get our money from the government, some from industry, although not for this. Motivations, I used to be in the Army, which is how I ended up interested in the topic. Just an overview, right? So your complement system is active after trauma. It generates a lot of inflammation. And fibrinolysis may be a key initiator of complement activation, but it's poorly studied, at least not directly. When we treat trauma patients with TXA, which is becoming ubiquitous in a variety of settings, it may have context-dependent effects on complement protein activation, whether it's the size of the dose, because I think you heard talks earlier about dosing and what's done and why it matters or doesn't matter. Timing, when it's given, right? We heard a lot about timing. And then we'll look at some data we've generated from the patch trial patient samples, as well as the Schreiber TXA TBI trial, and then non-TXA treated but well-described patients from Gene Moore's group. So the one thing I took away from the week is that the leading causes of death depend on who you talk to. So I'm going to tell you it's trauma and not heart attacks. But, up for debate. It's expensive. And coagulopathy, right? So people talk about coagulopathy and trauma a lot. A lot of times they don't talk about fibrinolysis, which is, I feel like, a large gap in thinking. Obviously not to this group, but for your broader audience. And there will be a bunch of background slides just to kind of get people up to speed, because personally, if somebody jumps into 40 slides of data and I know nothing about the topic, it's actually really hard to understand what they're talking about. So bleeding accounts for deaths like 25-ish% of the time. When they die really early, and if they die early and it's not of that, it's usually of a catastrophic brain injury. But about half the other deaths are in a delayed fashion, and I lump a lot of these into what I call the inflammatory basket, so organ failure, things like that. Fibrinolysis, right, we've heard a lot about that this week, and I'm just going to correct some terminology, because historically we've talked about fibrinolysis as hyperfibrinolysis, physiologic or non-pathologic, and shutdown, but it's really important, and Hunter Moore and Gene and me and a few others published last year, and then Liz Maginot, my resident, who you'll hear from soon, has a paper coming out shortly, where it's just a really key point, and it'll matter for this talk in a little bit, that if you look at active pine-1 and alpha-2 antiplasmin in a hyperfibrinolysis patient, not shockingly, those levels are much lower than in a hypofibrinolysis. But hypofibrinolysis and shutdown are very different groups. Shutdown had a lot of fibrinolysis. They have no inhibitors. And in fact, if you look at plasmin antiplasmin complex levels, they're a little bit lower, but still compared to hyper, but still much higher than hypofibrinolysis. And TPA pine-1 complex reflects a similar, although not perfectly same phenomena. Right, and this is important because we're going to look at complement in the context of fibrinolysis, okay? So when I say shutdown or hyper, they're different things than hypo. And if you correlated your free TPA activity and plasmin antiplasmin complex with TEG, this is just an interesting finding, again, for background. Our TEG, our regular TEG, is basically a random number generator. Unless you're really hyperfibrinolytic, you actually have to challenge it with TPA to see what's going on. Final few background slides. So complement, right? I told you it's active in traumatic injury. Does it matter? Well, you can basically discriminate survivors from non-survivors in a multiple different studies have shown that the people with the highest levels of complement activation die and have organ failure. And whether that is reflective of they're just sicker or is actually causative is hard to answer, because a lot of these things are associative, and unfortunately I think a lot of my data is that, but it's hypothesis generating for treatments. So we also know that of great interest, Marcus Huber Lang, a really smart orthopedic surgeon, and I hate saying that, but it's true, he figured out that you can generate a lot of C5 in the absence of C3, And in his initial model, it was felt to be due to thrombin, but subsequent work by postdoc in his lab showed that there's actually a hierarchy, like factor 10A, plasmin thrombin, 11A. They can all cleave, and 9A can cleave both C3 and C5, and they just have a slightly different order of efficiency. But plasmin is good at both. And I showed previously, taking in a layer of the onion deeper, that in trauma patients, this was a collaboration with Gene and with Jason Sperry at Pittsburgh, who ran the STAMP trial, that if I did a lot of neutrophil priming work years ago, and in panel B, if you showed a naive human neutrophil trauma plasma, in blue, they generate a ton of ROS if you give them a second stimulus, like with a formal peptide, which you would see when your mitochondria explode after a trauma. And if you treated them with a C5a receptor inhibitor in red, you abrogated the phenomena to the point where it looked a lot like showing healthy plasma to a human neutrophil. And we did this in a whole bunch of trauma patients and basically showed that you prime your human neutrophils for an inflammatory reactive oxygen species burst in a complement-dependent manner in trauma. So, and we showed that if you, these are ESIS experiments, that if you show neutrophils, product of the clotted blood, so serum versus plasma, with or without complement inhibitors, or with or without a poison of the NADPH oxidase called DPI, and then you put them in a transwell above the endothelium, that in fact neutrophils in response to blood clotting will release a lot of soluble mediators that generate degradation of barrier function of your endothelium, and you can rescue that by inhibiting complement receptors, or alternatively by poisoning the oxidase, so it's an ROS-dependent phenomena. And in a knockout mouse model, mice with chronic granulomatous disease, so their NADPH oxidase doesn't work, that they're protected from injury after trauma with shock, and we think that this is all complement-mediated, or a lot of it. So what about treatment, right? So I'm going to show you that fibrinolysis matters for complement activation, and when we treat people with TXA, it may be real context dependent, because we know, and I think most people in this room know, that when you give TXA in an idealized system, it will inhibit plasmin generation in response to TPA, but conversely, as was shown in the 80s and subsequently, when urokinase predominates, TXA actually has the opposite effect on plasmin generation, but what that does to fibrinolysis and complement is a little bit more unclear. So my opinion is that treatment with TXA probably does matter and influence these things, and one goal of mine has been to prove that, although I'm struggling in some regards. We'll see. So the hypothesis, right? Plasmin is known to be the most efficient coagulation-related, probably not the best term, but protease in the system at cleaving complement protein C5, and it's the second best at C3. And we know that complement is a bad actor in trauma, and that TPA is released in large quantities after trauma, and at least in animal models, and hopefully the patch trial, and Rob will be able to answer what happens to urokinase later after trauma. In animal models, especially TBI, it does get elevated at later time points and peaks at around 8 hours, right when we're finishing that life-saving drip. Not sure that's a great plan. And so we hypothesize that TXA inhibits plasma-mediated complement activation when TPA predominates, and at later time points when things like urokinase may be around, it may actually have the opposite effect. So initially we did this ex vivo, right, and we just showed that from purple to blue to red, not shockingly, right, you add more TPA to a blood clot, and on a turbidity assay, it lyses faster. And then if you look more importantly at the inset panel, right, from purple to blue to red, as you add TPA to clotted plasma, your complement activation goes up, and if you add TXA in the hash marks at physiologically relevant concentrations, you get significant, albeit partial, reduction in the complement that you activate. Okay, idealized ex vivo stuff. And if we depleted plasminogen on a lysine column, you lose that response to TPA where you don't get a burst in complement, but if you depleted C3, you keep it, and TXA inhibits that, so this demonstrates that it's a plasmin-mediated phenomena. And if I actually took those supernatants and showed them to neutrophils in red, you can see that if I lyse a plasma clot with TPA and show it to a neutrophil followed by a formal peptide, you get a big increase in ROS. If I showed it an anti-C5a receptor antibody, you reduce it, and if I show it TXA in blue, you also reduce it, albeit not quite as much. So you can inhibit complement-mediated neutrophil activation with TXA in the presence of TPA. And then when urokinase is around, urokinase, when TXA is around, does not lyse clots, at least not in the short term, although as TXA falls off, plasmin may be able to, as TXA falls off plasmin, it may happen. But what you can see is, if you clotted plasma and you add urokinase instead of TPA, you get some increase in complement activation. If you add TXA, it goes way up, right? That's consistent with our known phenomenon that you get more plasma, and actually it's independent of clotting. If you just add urokinase to unclotted platelet-poor plasma, nothing happens, but if you add urokinase with TXA, you get a big burst in complement, and so TXA does the opposite, and that was the underlying reason for our hypothesis that at later time points, TXA may actually increase complement activation. And it was the same thing when you depleted plasminogen, you lose it. When you deplete C3, you don't. So that's the interim summary in a beautiful picture that we published a while ago. But that's all ex vivo. So what about in vivo? And that's what really matters, right? And we're only about 11 minutes, and we got good time. So basically, TXA could inhibit or promote complement activation depending on which PA was around, but that's all ex vivo. Again, we need to test this in people, and it may depend on their injury status, et cetera, whether or not it works out. So big picture hypothesis, right? We think that, again, TXA may inhibit plasma-mediated complement activation when TPA predominates, so these early time points after injury, but later on, maybe that's not the case in our approach. We initially wanted to show that it had not been shown in humans, at least not convincingly, because as far as I know, we're the only group who published before that fibrinolysis and complement seemed to track together, but it was a very limited data set we published before. So we looked at 56 well-described trauma patients that Gene Moore was kind enough to share the plasma with my group, and we looked to see whether or not fibrinolysis tracked with complement activation. And then we got another 56 patients from the patch trial, complements of Rob and Russ and Dom. And then we got another 40 patients from Marty Schreiber's group, which is the US TXA TBI trial. And they're slightly different. We got different doses so that we could evaluate a dose response, although the patients are a little bit heterogeneous between the trials. And we evaluated complement as a function of fibrinolysis. So part one, does fibrinolysis track with complement activation in trauma patients who are not treated with TXA who have a well-described fibrinolytic phenotype? Median age is 34. In most trauma trials, you'll see that it's 70 to 80% men because we just can't help but do dumb things. And in this cohort, it was 50% penetrating, I believe predominantly gunshots. It's a good advertisement to visit Denver. And then the NISS was 14 with kind of a wide IQR because some of them were really hurt and some were not. And this is on Liz's poster in the back, but I've cut the range bar because there's like a couple of outliers in each, so you can't really appreciate back there the median difference. If you look at C3A, right, this is hypofibrinolysis, hanging around 100 nanograms per ml, non-pathologic, about the same value, and you get a couple of weird outliers. I'm not sure what that is. I haven't looked further. But in hyperfibrinolysis, your median, right, this middle line in your box and whiskers is threefold higher than in hypo and non-pathologic. And in fibrinolysis shutdown, remember I told you they had fibrinolysis, it turned off, so they're not actively lysing, but they're like a haircut away from a bleed, has an increased but not as much complement activation. And this really kind of cues you in that over the course of fibrinolysis, you increase your complement activation. When you turn it off, it comes back down. You see the same thing with C4A, which is a little bit interesting. I'm surprised by that because I'm unaware of plasmin and other coagulation proteins cleaving C4. We could go deeper into why. I think it's probably a lectin pathway thing, but I can't prove it yet. C5 has the shortest half-life of all of them. You do see the same changes in medians by and large. It's up in hyper and kind of middle of the road in shutdown, but not as good. And I think it's probably a half-life related phenomena because in the longer half-life version of C5, right, where soluble C5b through 9 it sticks around a lot longer, you see the same phenomena you saw with C3. So hyperfibrinolysis in polytrauma patients was strongly associated with activation of the complement system. And in keeping with that, if you look at the complement control proteins, right, factor I, which is a protease that cleaves C3b and C4b, both in solution and the cell surface, is exhausted in your hyper and your shutdown patients, but preserved in your hypo and non-pathologic. And the same is true of factor H, which is a cofactor for factor I, and also a cofactor for decay accelerating factor. So it all fits the story, right? And that made it worth pursuing, looking at Patch and looking at Marty Schreiber's data. And just really quickly, if you correlated, and this is, you probably saw this, if you correlate the complement activation fragments and the complement control proteins. They track really well, your activation markers with markers of activation of fibrinolysis and inversely with your, inhibitors or exhaustion, and so it all fits the picture. So part 2, does pre-hospital TXA with a 1 gram drip plus bolus from the patch trial influence complement activation? So we got 56 patch trial patient samples that had plasma in the ED and at 8 hours and at 24 hours. 53 of them were a blunt mechanism, so to avoid heterogeneity and outliers, we excluded the 3 penetrating trauma patients, which is standard practice when you're trying to look at these groups. Median age was 40, again, mostly dumb men. The ISS was much higher. These were really sick patients. And I think it has to do with what the enrollment criteria are with their COAST score. It just sort of naturally selects for this. On univariate analysis, so anytime you get a subgroup from a trial, right? So this was a 900 a large trial, and then we only got a subgroup plasma samples. You just have to make sure that randomization was preserved. On extensive analysis, the only thing that actually came out was temperature. It was a degree Celsius difference between the groups, which is clinically insignificant. So they were well preserved. And we did complement multiplex, not on a bead system and not on, like aptamer assays or anything, because I found that those don't actually track well with validated assays. So this is essentially a system by Quidel, which is a company that does a lot of complement stuff. That it's essentially an ELISA, but it has microdots on a 96-well plate. So it helps you kind of with your throughput, looking at activation and regulatory markers. And then it does not evaluate, obviously, plasma antiplasmin or your lectin pathway proteins. So I did those separately. And because our hypothesis is biologically different between early and late time points, normally when you look at changes over time, things like linear mixed models, when you have 3 or more time points are appropriate, but because our hypothesis shifts between ED and then the 8 and 24-hour mechanism, you end up getting a really noisy signal that may not, you'll get biologically incorrect conclusions from these types of models. We actually intentionally did not do that here. So they're just pairwise comparisons based on early and then the later 2 time points. And in summary, at least in the 1 gram bolus plus 1 gram drip patients, we did not see any differences between TXA and not at early time points, right? So on C3A, they look the same. C5A, they look the same. Solid with C5B through 9, big error bars, they basically look the same. At the later time points, though, what was of significant interest to me is that at 24 hours in the no TXA versus the TXA group, you got a pretty big increase in C3A. It's like 1.6, 1.7 fold. It's quite a bit. And C5A did the same thing, actually. It went up and significantly. So soluble C5B through 9 did not. I think this is, again, likely related to half-lives. And if you look at, there are some limited series of complement activation time series in trauma where it's seen ED 24 hours and then every day for the next 10 days. And you get a couple of pretty well-described peaks, seeing ED 24 hours and then like 7 to 10 days and everything in the middle, it's back down. So I wonder if it may be reflective of that. But the bottom line is our short half-life antiphyllotoxins demonstrate an increase at a later time point. I can't tell you it's because of urokinase. I would think it is, but obviously this is associative and not causative. But if it's at least part of our hypothesis, I'm a little surprised by the early result, though. So if you looked at the alternative pathway, right, BA, and the reason I use BA and not BB, I have both, is that it was in the dynamic range of my assay at the dilution I was using, like dead center, whereas BB was at the extreme, so I kind of don't believe it as much. And then we looked at plasma antiplasmin, and your alternative pathway, I've shown and other people have shown it's activated after a complement. It's a little weird. I don't know why it's so much more active at 24 hours, but it is, and it's significantly up, but it's not different between the groups. Plasma antiplasmin, don't take this as gospel values. I don't want to draw any conclusions about PAP from what I did, because it was just a single ELISA that may not be up to snuff with most in the room. But at least in my hands, there's not a difference between TXA and no TXA in the plasma antiplasmin level at any of the time points between the groups, although not shockingly, the level consistently goes down as time goes on from time of injury. That's not a surprise. Interesting findings to me. And when I looked at MASP and mannose-binding lectin, I don't see anything that sticks out to me. The one thing I do want to do, though, is look at quartiles of patients, because these are all the patients lumped in a group, and I think when you discriminate them by quartile, you actually find some interesting biology. But as an entire group, no different. And you'd actually be expecting, you see a decrease in mass consistently, but obviously not even close to significantly in the TXA group. That, to me, is worth following up. I'm not going to pontificate on it too much other than to say that TXA may be a shed ACE inhibitor, and we've talked about that. It inhibits your release of or glycocalyx shedding. And a project I'm working on, I don't have data to show here, is looking at whether or not your lectin pathway, which some have shown is active in trauma, is actually being activated by glycocalyx components that are shed, and TXA may actually inhibit that. And I'll follow up on that later. Mannose-binding lectin, we're actually about 20% of us in this room are deficient in it, which is why you get these massive error bars, and it makes it really hard to actually generate any real data unless you knew who was deficient and then exclude them from your analysis. It's up there, so in full disclosure, we've looked at it. And factor I and factor H, it was preserved in, factor I in particular was preserved in the TXA versus no TXA group across all time points, but not significantly so. It may just be random, and the differences aren't large. So what happens if you correlate plasma, antiplasmin, and complement? So in the emergency department, it's kind of a random, just a shotgun of stuff, but the few things that stood out to me, and I'll go through all the time courses really quick. It's kind of hard to look at this presentation, but it's kind of a weak correlation between PAP and soluble C5b through 9, and it's not significant. But if you just look at hypotensive patients, right, the patient with blood pressure below 90, all of a sudden C5a, 0.64 for a correlation coefficient, just about 0.5 for soluble C5b through 9. So in patients generating a lot of plasmin in a hypotensive patient, it appears that they may correlate reasonably well. And there are just a couple other things, and you'll see it because I'll blow through the next two, are that your alternative pathway tracked really well with C5 activation fragments across all time points, as actually did C4a. Not surprising because they're upstream from those generations in most cases. But and we looked at it by TXA status, and the only thing that really stuck out to my eye is that at 8 hours, for whatever the reason, once you analyze it based on whether you got no TXA or got TXA, all of your C3 and C5 components did actually correlate with plasmin and antiplasmin, which I'm not sure what to make of it yet. But the punchline is that at early time points, there appears to be no impact at this dose of TXA or dosing regimen on complement activation, but later on it appears to increase complement activation. And your alternative pathway is activated later, but does not appear changed by the TXA status. And plasmin and antiplasmin strongly correlated with C5 cleavage in hypotensive patients in the ED. And at 8 hours, your plasmin antiplasmin level, when you discriminated based on TXA status, seemed to correlate reasonably well with a lot of your activation markers. Your alternative pathway in C4 correlated with C5 activation across all time points. And to me, the alternative pathway is really an amplification pathway. So chasing down whether lectin or classical is involved is important in the future. So I've only got a couple minutes. I'll just wrap it up and say we then looked at Marty Schreiber's trial. We got 40 patients. Only 60% of his trial patients ended up having a confirmed TBI. I wanted to only look at those with a confirmed TBI. So we got 20 with a confirmed TBI who got 2 grams of TXA bolus in the field, 2 grams or placebo. And an important point of his trial is that his trial excluded hypotensive patients because they wanted the altered mental status, the GCS of 12 or less, to be reflective of just that and not, hypotension. An important finding in their trial was that there was no difference in bleed size over serial cross-sectional imaging between the groups. And so it begs the question, there was a mortality benefit to the 2-gram pre-hospital bolus, but the bleed size was not different. And so what's going on? And if you look at animal models, if you give them an intracranial hemorrhage and then you provide them with complement inhibitors, they have less inflammation in the brain, they have less vasogenic edema, and they do much better on multiple different functional outcome scores over time. And so to me, it was consistent with maybe TXA as having an anti-complement effect and preventing secondary brain injury from edema. So we did the same stuff essentially, and again, mostly men. High median AIS had, preserved randomization. They were fairly well-matched groups. And we get the total opposite response, actually. So in this case, a couple things worth pointing out. So first, C5a is significantly reduced at an early, or at the late time points. So we don't we don't see a significance at the early time point, although there's an obvious difference between the groups, but you do hit significance later on. But of interest, right, soluble C5b through 9 is about half as much in the TXA group compared to no TXA. And you see this kind of creeping increase, though, it's even higher in the TXA group later, which does not match this. I think, again, half-lives versus assay random error. But what caught my attention is this. Look at the error bars in your C3A in all of your no TXA groups, which are much smaller in your TXA groups. And it was suspicious for me, so then I looked at median and IQR and median and range, and you see that your top quartile of patients in the placebo group is basically represented by this group, is markedly different than your TXA group, and you don't have any of those outliers in the TXA group. You just never get that high. So we're going to look at the quartiles and see what's different between the patients. I'm having a minor disagreement with our statistician about its importance, but I think without chasing it down, you won't get to the biology. Plasmin, antiplasmin was trending lower in all the TXA groups early on and flipped towards the later time points. I didn't do this assay, Marty did them, but D-dimer was lower, essentially, always in the TXA groups, although not always significantly. And factor H was preserved at that early time point, consistent with less complement activation. So at least at the early time points in the 2-gram bolus of TXA, we did show a significant reduction in some of your complement activation markers and preservation of one of your complement control proteins. So it appears to significantly reduce cytotoxic soluble C5b through 9, which has a longer half-life, so it's more of a global readout of what's been happening, but that can also confuse the picture. And we're looking at that top quartile of C3a, and that's actually the soluble C5b through 9 has a similar looking phenomena. See what's different about those patients. It may dampen early vasogenic edema, and while those late time points you start to get kind of a messy picture, I think what happens early on sets the stage for what happens later in a lot of these patients. And happy to take questions. I have too many children, and they're all young, and thanks to my collaborators.